Have you ever looked at an NMR spectrum and felt like you were staring at a secret code? Peaks everywhere, numbers that don't make sense, splitting patterns that look like hieroglyphics. Don't worry, you're not alone. Today, I'll break down nuclear magnetic resonance spectroscopy in the simplest yet most detailed way possible. By the end of this video, you'll not only understand what those peaks mean, but also be confident enough to interpret spectra like a pro. So, grab your notebook, because this is going to be the ultimate guide to NMR spectroscopy.
Alright, so you've probably seen those crazy-looking machines in science labs with big coils and warning labels and wondered, what exactly is NMR? Let me break it down. NMR stands for nuclear magnetic resonance. Sounds intense, but the principle is surprisingly simple, and kind of magical. At the heart of it all are tiny particles called nuclei, which are just the centers of atoms. Some of these nuclei, like the ones in hydrogen atoms, act like little magnets. Yup, each one is spinning and generating a magnetic field. Now, if I place a bunch of these spinning nuclei in a really strong magnetic field, way stronger than your fridge magnet, they all line up. Some point with the field, some against it. Think of tiny compass needles trying to align. Here's where it gets cool. Hit them with radio waves at just the right frequency. They get excited, flip orientation, like tuning into their favorite radio station. Turn the radio wave off, they relax back, and release a tiny signal. That tiny signal is what we measure in NMR, like eavesdropping on secret conversations of atoms. Pick up those signals, and you can tell what atoms are in a sample.
Okay, now the question is, how do we read these signals? The first and most important concept is the chemical shift. This is measured in parts per million, ppm, and tells us about the electronic environment around a nucleus.
So chemical shift is basically a way to tell how shielded or de-shielded a nucleus is by its surrounding electrons. Think of it like sunblock. The more electrons hanging out around a hydrogen atom, the more protected it is from the external magnetic field. Less sunblock? That hydrogen feels the force more, and its signal pops up further downfield, so a higher chemical shift. Now, in an NMR spectrum, each unique hydrogen in your molecule shows up at a slightly different spot, because each one sits in a unique electronic neighborhood. It's like every hydrogen has its own little zip code. Hydrogens stuck to carbons next to electronegative atoms, like oxygen, get pulled away from their electron cloud sunblock, so they show up at higher shifts. Hydrogens stuck to carbons next to electronegative atoms, like chlorine, get pulled away from their electron cloud sunblock, so they show up at higher shifts. That's why you'll see alcohol hydrogens at different positions on the chart. That's why you'll see hydrogens near double bonds at different positions on the chart. By looking at where these signals land, chemists can piece together a map of the molecule, almost like solving a puzzle.
Next, we need to figure out how many hydrogens are responsible for each signal. This is where integration comes in. The area under each peak is proportional to the number of protons. Take ethanol, CH3CH2OH, as an example. The methyl group, CH3, shows up as a peak with 3 hydrogens worth of integration. The CH2 group shows up with 2 hydrogens. And the OH proton, just 1. Add them up. That's 6 hydrogens matching the formula of ethanol. Boom! Now you see how NMR helps confirm molecular formulas.
Here's the trick. Integration. In NMR, integration means measuring area under each peak. That area is proportional to the number of hydrogens. If one peak's integration is twice another's, that means twice as many hydrogens created. Let's say you've got 3 signals with integrations. Integration equals 3 hydrogens for the first signal. Integration equals 2 hydrogens for the second. Integration equals 1 hydrogen for the third. Pretty cool, right? But hold up. Integrations don't always give whole numbers. Sometimes you'll see weird fractions or decimals. Don't panic. Find the simplest whole number ratio between the signals. That ratio tells you how many hydrogens each set represents. Pro tip: integrations don't tell total hydrogens without the formula. They do tell relative numbers, so you can piece together locations. In a nutshell, integration is your NMR cheat code. It removes guesswork for how many hydrogens lie behind each peak, so you can solve structures faster and with more confidence.
Now comes the fun part, splitting patterns. Protons don't exist in isolation, they feel the magnetic influence of their neighbors. This gives rise to multiplets. The rule is simple: a proton with n neighboring equivalent protons splits into n+1 peaks.
NMR secrets. Master the n+1 rule instantly. Alright, so you've just started learning NMR, and you keep hearing about this n+1 rule for spin-spin splitting. Sounds mysterious, right? Let me break it down, step by step. Picture this. In NMR, each hydrogen feels the magnetic influence of its neighbors. If a proton is next to n other hydrogens on adjacent atoms, its NMR signal splits into n+1 peaks. That's the n+1 rule. Let's make it concrete. CH2 next to CH3. 2H and CH2 beside 3H and CH3. So CH2 splits into 3 plus 1 equals 4 peaks. Quartet. CH3H next to 2H from CH2, so CH3 gives 2 plus 1 equals 3 peaks. Triplet. Why? Each neighbor can align with or against the field, creating slightly different environments. More neighbors, more combinations. Not every neighbor counts. Only hydrogens on adjacent carbons. Don't include hydrogens on the same carbon or ones separated by more than 3 bonds. See a doublet-triplet quartet, you'll know. It's just the n+1 rule in action. Count the neighbors, add one, boom. Once you get this, interpreting NMR becomes way less scary.
Alright, let's break down the 1H NMR spectrum of toluene, also known as methylbenzene. This is a super common molecule in organic chemistry, and its NMR is a classic textbook example. First, let's talk structure. Toluene =sign benzene ring plus single methyl group. In 1H NMR, we look at hydrogen environments. 2 main types of hydrogens. 5 on the benzene ring, 3 on the methyl group. Starting with the methyl, 3 equivalent hydrogens next to the ring. They show up as a singlet, usually around 2.3 parts per million. Now the aromatic hydrogens, a bit more interesting. 5 aromatic H's are not identical due to the methyl, but the differences are pretty subtle. Aromatic protons appear as a multiplet, around 7.07.3 ppm. That's from splitting by neighbors on the ring. Summary: singlet for methyl H's, and a multiplet for aromatic H's.
Let's take one more example to understand more about NMR.
Isopropyl alcohol, or CH3-2CHOH, has 3 types of hydrogen environments. 2 identical methyl groups, those CH3ES, show up as a big doublet signal, split by their neighbor, the single CH on the middle carbon. That central CH, it pops up as a multiplet thanks to the 6 hydrogens next door. And the OH group? It's usually a singlet appearing all by itself.
So why is NMR so important? Because unlike other techniques, NMR doesn't just tell you the formula, it tells you the exact arrangement of atoms. It's like having X-ray vision into the molecule. That's why NMR is a central tool in organic chemistry, pharmaceutical industry, and even in biology for studying proteins and DNA. Now, I want to hear from you. Which part of NMR do you find the most confusing? Chemical shifts, splitting patterns, or advanced spectra? Drop your answer in the comments, and I might make a dedicated video on that. If you found this video helpful, don't forget to like, share, and subscribe for more deep dives into chemistry. Until next time, keep exploring the molecules around you.